Welcome. Today, we are going to examine why most students fail in Section C of the CSIR NET Life Sciences examination, despite solving thousands of questions and attending expensive coaching programs. The core problem is not the volume of questions solved, but the way students learn from them. Conventional preparation relies heavily on memorization and pattern recognition. Coaching often explains what the answer is, without teaching the underlying reasoning process. Excessive mock tests do not build mechanistic understanding or analytical frameworks. To score high in Section C, you must move beyond pattern matching and develop genuine analytical and quantitative thinking. This is precisely what is needed, and it is exactly what the JRF Academy design of MCQs provides. To address these shortcomings, we must adopt a different learning philosophy. The design of our MCQs at JRF Academy provides exactly what is needed for scoring high in Section C. We do not train you to recognize familiar questions. We train you to solve unfamiliar ones. Every MCQ is structured as a complete learning experience that builds 6 core cognitive skills: conceptual understanding, analytical reasoning, quantitative thinking, logical deduction, scientific problem solving, and adaptive thinking. By focusing on these core skills, our platform provides the exact mental framework required to master the most challenging questions in the examination. How exactly does the JRF Academy MCQ design provide this? Each question functions as a self-contained learning unit with 7 integrated layers that progressively rebuild your mental model. First, the core concept. Second, the mechanistic basis. Third, step-by-step reasoning. Fourth, distractor analysis. Fifth, visual encoding through scientific diagrams. Sixth, concept mapping to link adjacent topics. And seventh, new question application. This structured, adaptive learning methodology is what is needed for scoring high in Section C, systematically replacing rote memory with deep analytical frameworks. Now, let's look at how this works in practice. Let us begin with a quantitative question from Unit 1. What is the question actually asking? We must calculate the reaction velocity of an enzyme with a given Km and Vmax in the presence of a competitive inhibitor. The biological mechanism tested is Michaelis-Menten kinetics modified by competitive inhibition. Why do many students answer this incorrectly? They apply the standard velocity equation without adjusting for the inhibitor, get a value not listed in the options, and panic guess. Approach this analytically by calculating the inhibitor's effect. The clue is the specific inhibitor concentration and key. Signaling an adjustment is mandatory. A competitive inhibitor raises apparent Km by a factor alpha, calculated as 1 plus I over key. Here, alpha is 3. Multiply alpha by the original Km of 2 millimolar to get an apparent Km of 6 millimolar. Plugging this into the velocity equation, 100 times 4, divided by 6 plus 4, yields 40 micromoles per minute. The concept explanation deepens understanding by demonstrating how the inhibitor alters enzyme affinity without affecting the catalytic ceiling. The visual diagram reinforces this by showing the curve shifting rightward. Apparent Km increases. Vmax remains the same. The concept map organizes these relationships, contrasting competitive with non-competitive inhibition. How can you transfer this reasoning? By applying the exact same mathematical logic to new questions on drug IC50 interpretation or metabolic flux analysis. Next, a conceptual question from Unit 2. What is the question asking? A protein destined for the inner mitochondrial membrane is arrested within TIM23. We must identify the structural feature causing this arrest. The concept tested is the TIM23 import pathway. Students answer incorrectly because they confuse the N-terminal matrix targeting sequence with the stop transfer signal. Analytically, look at the protein's final destination. If it is arrested and inserted laterally, the signal must be hydrophobic. Option A, the amphipathic targeting sequence. drives full matrix translocation, not arrest. The clue is lateral membrane insertion, pointing directly to a hydrophobic stop transfer segment. Option B. The concept explanation reveals TIM23 operates in two modes, a translocase channel and a lateral gate. The visual diagram reinforces this by showing the hydrophobic transmembrane domain stalling within the complex. The concept map organizes these relationships by linking protein topology signals to their specific membrane insertion pathways. Transfer this reasoning to solve completely new questions on ER signal anchor sequences or clinical mutations causing diseases like MERRF. Here is an analytical question from Unit 3. The question asks, if rifampicin is added after RNA polymerase has synthesized a 15-nucleotide RNA chain, what happens to the transcript? The concept tested is the specific mechanism of rifampicin. Students answer incorrectly by assuming antibiotics act throughout a process, confusing rifampicin with elongation inhibitors. Analyze the drug's target and transcription stage. Rifampicin blocks initiation, but the critical clue is the 15-nucleotide chain. Promoter clearance occurs around 9 to 12 nucleotides. At 15 nucleotides, the polymerase is already elongating. In an elongation complex, the rifampicin binding site is occluded. This clue eliminates options suggesting immediate halting or degradation. Transcription continues to completion. The concept explanation clarifies the stage-specific nature of this inhibitor. The visual diagram reinforces this by showing structural strict hindrance at the binding pocket once the RNA chain extends. The concept map organizes inhibitors into three classes: initiation blockers, elongation blockers, and topoisomerase inhibitors. Transfer this framework to new questions regarding stage-specific pharmacological interventions in any biological process. A comparative question from Unit 4. The question asks how EGF and glucagon, acting through structurally distinct pathways, both activate the same transcription factor. The concept tested is signal convergence. A common error is assuming convergence requires a shared intermediate, leading students to select a single shared kinase. Compare the pathways analytically. EGF acts via receptor tyrosine kinase TERK. Glucagon acts via G protein to protein kinase A. They are distinct. The clue is that both activate a common target. Their respective kinases can phosphorylate the same transcription factor, CRED, eliminating options requiring a shared upstream mechanism. The concept explanation illustrates how networks use distinct routes to modify common downstream nodes. The visual diagram reinforces this by showing the two distinct kinase cascades converging on the same target. The concept map organizes these relationships by linking ligand-receptor specificity to downstream effector redundancy. Transfer this reasoning to new questions on therapeutic resistance, where blocking one pathway is insufficient because a parallel pathway still activates the target. An integrative question from Unit 5. The question asks why ablation of the P4 blastomere in C. elegans results in no germline, and what accounts for cell autonomous restriction. The concept tested is cell autonomous specification. Students frequently err by selecting inductive signals, because C. elegans is famous for inductive signaling. Note the phrase cell autonomous. If P4 autonomously becomes germline, it must inherit determinants blocking somatic fate. Option A. Inductive signals contradicts cell autonomy. The correct mechanism is option B. P4 inherits cytoplasmic determinants like PY1, repressing somatic transcription. The concept explanation highlights that PY1 represses RNA pol2, silencing somatic genes. The visual diagram reinforces this by showing P granules asymmetrically segregating into the P lineage. The concept map contrasts cell autonomous determination with inductive signaling. Transfer this exact reasoning to new questions on muscle determination via MYOD, or neurogenesis via lateral inhibition. A factual question from Unit 6. The question asks which mechanism primarily suppresses photorespiration in C4 bundle sheath cells. The concept tested is the C4CO2 concentrating mechanism. The common error is selecting that bundle sheath chloroplasts lack photosystem II, while some C4 species are photosystem II deficient. This alone does not eliminate photorespiration. Consider the kinetics of RuBis-CO. Photorespiration occurs when oxygen outcompetes CO2. The correct answer is that decarboxylation of C4 acids concentrate CO2, outcompeting oxygen. This clue shifts the carboxylase to oxygenase ratio favorably, eliminating the photosystem II option. The concept explanation clarifies that C4 plants solve this kinetically by altering local substrate concentration. The visual diagram reinforces this by showing spatial separation. PEP carboxylase in mesophyll, and malate decarboxylation raising CO2 in the bundle sheath. The concept map links anatomy, biochemistry, and kinetic competition. Transfer this framework to solve new questions on crop engineering, like introducing C4 traits into C3 plants. An analytical question from animal physiology. During sustained exercise, an athlete shifts from glucose to fatty acid oxidation. What explains this switch? The concept tested is AMPK-mediated metabolic switching. Students answer incorrectly by selecting that increased insulin promotes GLUT4 internalization. This is flawed because insulin levels fall during exercise. Focus on cellular energy sensing. The clue is sustained exercise, implying ATP depletion and a rising AMP to ATP ratio. This activates AMPK. AMPK phosphorylates and inhibits acetyl-CoA carboxylase, reducing malonyl-CoA. Since malonyl-CoA inhibits CPT-1, its reduction relieves CPT-1 inhibition, allowing fatty acids to enter mitochondria. This eliminates the insulin option. The concept explanation reveals the AMPK, acetyl-CoA carboxylase, CPT-1 axis as a core regulatory hub. The visual diagram shows the stepwise molecular cascade from AMPK activation to fatty acyl-CoA transport. The concept map organizes relationships between energy state, allosteric regulation, and substrate transport. Transfer this reasoning to new questions on type 2 diabetes, where metformin activates this same pathway. A quantitative question from inheritance biology. Cystic fibrosis occurs in 1 in 2,500 births. A confirmed carrier woman marries a man with no family history. What is the probability their child has CF? The concept tested is Hardy-Weinberg equilibrium applied to clinical risk. Students err by selecting 1 in 2,500, citing population frequency, or forgetting the 1 in 4 Mendelian probability. Condition on the mother's status analytically. The mother is a confirmed carrier, so her probability is 1. First, calculate the father's carrier probability. If Q squared is 1 in 2,500, Q is 1 in 50. The carrier frequency, 2pq, is approximately 1 in 25. Second, if both parents are carriers, the probability of an affected child is 1 in 4. Multiply these. 1 times 1 in 25 times 1 in 4 equals 1 in 100. The clues force you to reject the population average. The concept explanation deepens understanding of integrating population genetics with Mendelian inheritance. The visual diagram shows the Punnett square intersecting with the population curve. Transfer this quantitative reasoning to new questions on sickle cell anemia or Tay-Sachs risk estimation. An integrative question from Methods in Biology. An RNA-Seq experiment identifies 847 differentially expressed genes. To determine if they form a coordinated program, which strategy is most informative? The concept tested is multi-layered bioinformatics validation. A common error is selecting individual RT-qPCR validation for all genes. This only confirms differential expression, not coordination. Analytically, a coordinated program requires functional, regulatory, and physical interaction evidence. Option B combines gene ontology enrichment, transcription factor binding motif analysis, and protein-protein interaction networks. Gene ontology tells you what functions are enriched. Transcription factor motifs tell you what regulates them. Protein interaction networks reveal if the encoded proteins interact physically. This eliminates single tool options. The concept explanation clarifies that systems biology requires integrating orthogonal data types. The visual diagram reinforces this by mapping isolated genes into a connected, multi-layered interaction network. The concept map organizes analytical layers from raw data to functional modules. Transfer this reasoning to design and interpret multi-omics pipelines for completely new questions in cancer biology. To make this methodology tangible, let us look at how visual encoding is applied in practice on the platform. After every question, students receive a professionally designed scientific diagram encoding the underlying concept, not just the answer. For example, when studying peptide bond chemistry, a diagram illustrating resonance delocalization instantly explains the structural basis of backbone rigidity. In protein DNA binding, visualizing cooperative stabilization shows how individual factors that bind weakly alone lock into stable complexes together. For gene regulation, enhancer-promoter footprinting diagrams reveal exactly where protein complexes bind and protect the DNA from cleavage. These visuals appear immediately after each question, building permanent scientific mental models rather than simply confirming a correct letter. In Section C, many questions embed data directly inside the prompt, requiring visual interpretation. Our platform integrates these exact challenges. For instance, in a time course footprinting question using DNA protection analysis, the student must read the gel and deduce whether a protein DNA complex is progressively stabilizing or dissociating across different time points. Similarly, in a competition assay, interpreting the return of bands as a competitor increases reveals the transient exposure of DNA and the mechanism of protein exchange. With over 21,000 analytically engineered questions, every diagram demands active reasoning, forcing students to interpret experimental data just as they would in a real laboratory setting, rather than recalling a memorized fact. Finally, after analyzing the question and studying the diagram, students receive a structured reasoning map. These maps encode the logical chain from experimental observation to mechanistic conclusion. Take phospholipid structure. The map traces the polarity of the PO bond to its high electronegativity difference, resulting in a strong dipole and water-soluble behavior. In a more complex scenario, like a SHP2 knockout in CAR cells, the map links normal CXCL12 secretion to an intact endosteal niche, thereby explicitly ruling out niche disruption as the primary cause. These reasoning maps are generated for every question, mapping each logical step so students internalize the scientific method itself. This brings us to our conclusion. The traditional strategy of memorizing thousands of questions simply does not work for Section C. A better strategy is to master concepts deeply. By learning through just 25 to 50 carefully designed questions per topic, you progressively build strong conceptual foundations, mechanistic understanding, logical and analytical reasoning, quantitative problem solving, and adaptive scientific thinking. Once these mental frameworks are established, you become capable of answering any type of question: factual, comparative, analytical, quantitative, or integrative, because you understand the underlying concept, not just the format. This approach prepares you not only to perform better in the examination, but to succeed in PhD interviews, laboratories research, and a long-term scientific career. Master concepts, develop scientific thinking, build your research career. Explore JRF Academy today at jrfacademy.com.